Bismillahirrahmanirrahim. Assalamu alaikum. This is Hafiz Muzammil Rahman. In today's tutorial, we will try to analyze the results of the docking by the Autodock tool, or if you have any complex, that is protein ligand complex or protein complex, so you want to analyze the result, or you want to make a high quality publishable image or high quality image which you want to publish in a research paper. So this tutorial will definitely help you. Let's do start it. We have docked our complex with the ligand in our previous video. So we will start from there. We are going to analyze, then docking, then open. So our DLG file will be here into our directory. So we will open it. So there are 10 conformation which are present in that specific DLG file, docking log file. So we will click on it, and now we will go to the analyze again, and then we'll click to the macromolecule, and then we'll click only open. So the molecule or the receptor that will be open. So now we have a protein ligand complex. So we want to analyze the results firstly in the Autodock tool, and then we'll move forward. First of all, you'll have to go to again. I'll change the little bit presentation of it, and here we are going to change the ligand. This is the ball arrangement. Yes, this is a little bit better. So this is our ligand now. So I'm going to change the structure or the audience or presentation of this receptor as well. So I'm clicking to the ribbon. This is R, below the R over here. It will take some seconds, and now okay, so this is our receptor and this is our dock ligand. Now we are going to again the analyze, and then we'll click on the conformation and then play. So here this window will show the different orientation of our ligand. So while clicking on this is second, third orientation, fourth one, fifth one, sixth one, seventh, eighth, ninth, and this is 10th one. So we can check how many hydrogen bonds this ligand is making with our receptor, or we can go to the DLG file directly and can check the energy and all other things. So the conformation 6, it has more energy, and it is making hydrogen bond with the receptor. So click on this window or this option, then there will be set play option window will open, and click on the show info. So this will show information regarding this specific orientation of the ligand, like the binding energy, that is minus 10.53, very good binding energy. Inhibition constant is 19.19 nanomolar, and internal energy is this, and all the information it is available in this small conformation box. And then you can check this one, build hydrogen bond as well. So check this one. Yes, so this is the window which is showing the hydrogen bond information, and here you can see these are the hydrogen bonds, and the distance and the bond length, they are shown like here. So it is making two hydrogen bond, that is 2.171 angstrom and 2.217 angstrom. So the next step is we are going to export this complex, because we will must have a complex if we want to analyze the results. So we are exporting this complex for the further analysis from the Autodock. So here there is option, that is write complex. click over here. Yes, so it is we are in the directory that is docking, or wherever you want to save this specific complex, so you can save it. So here we are going to save this by the name of docked. Okay, so the file format is pdbqt. So click on the save. It is showing I have already the complexes by this name because I have saved it. So we can write any name like new underscore complex. Okay, so new underscore complex. Yes, so it is saved in our directory now. Here, if you will open this docking folder, so it is showing new. Where is the n? Yes, this is new complex.pdbqt. Okay, this is new complex .pdbqt. So now we are going to analyze this complex in PyMol, and then we will export, or we will change the format from PDBQT to PDB, protein data bank. Okay, so where is the PyMol? Yes, this is the PyMol. Let me clear it, delete all. Okay, so this is the PyMol. I have changed the background of the PyMol from the here display. If you will open the PyMol, the background will be black. So you can go to here display and then background. So click this white. So that will be white. If you will click black, so when you will open it firstly, so the background will be like this black. So I'm again going to make it white. Okay, so I have changed the background to white because I want to analyze the result more clearly. So you can import the file from here open, or you can directly drag the file to here. I'm just going to drag it. So yes, so I have dragged the complex. The name of complex was, what was the name? The name was new underscore pdbqt. Okay, so this is the complex. This is the ligand. I'll click it, the ligand, that is change show into the sticks, that is already in the stick format. So you can change it like this. Okay, now I'm going to go to the action, or A, this tab A. Here there is the find. So in find, click polar contact, and in polar contacts check the any atom. I am repeating it again. If you have imported any complexes, that whether that is in PDB or PDBQT in PyMol, so I am going to show you the interaction between the ligand and the receptor. So if you have any other receptor, so you can find out the interaction in this way. Okay, go to A for action, find, then polar contacts, and then to any atom. So now these are the contacts in the yellow dots. Like, I'm just going to zoom it. Okay, these are the contacts, but there is a problem. In fact, this is not a problem. Actually, you don't see the interacting residues. Which are the residue by which your ligand is making the bond. So I am now going to solve this problem. Come here, show at the very beginning, S for show for lines. So now you have lines. So from here you can select the residue one by one like this. Just click on it. This residue has been selected, which is making hydrogen bond. And again, okay, let me change the display to black. Yes. Another one is this. This residue is making hydrogen is making interactions, and I'm little bit zooming out it. And yes, this one, this is making bond. Confirming it. It will be a little bit tricky, but you can do it easily. Like, okay, so which one is left? I think so, this one. I'm just confirming this. Yes, this is the residue. Okay, so again go here and show sticks. Okay, very good. So the residue which you have selected, they are shown into the stick conformation. Okay, so click at the empty place. Go again here. Sorry, in the start there is H for hide, and then hide the lines. Okay, so now the lines has been hided. Okay, so now you are looking the residues which are making hydrogen bond with this ligand. You can change the color as well, but my concern is not that. Okay, so these are the hydrogen bond. Now if you want only if you want only those residues which are making hydrogen bond or which are making interaction with your ligand, and you want to hide the all other, so in this pose you can make a very good picture. If you want to make a picture, or you want to make an image in this specific orientation, so you can go here and then export image as PNG. So while clicking on this, so it will save the image in this conformation. I'm going to save it firstly for you. Save like image 1. I'm giving the name to image 1, and if it is, this is my directory, docking, so that is saved. Okay, so this image is saved. If you want to analyze only those residue which have some interaction and you hide other one, so you can do it again from here. Come to the H and click. H mean hide. Click the ribbon. Okay, so these ribbon structure has been hidden. Okay, so now you have only the interacting residues and the ligand. If you want to label the distances, so you can do this as well. Just go here, come here and click S, and then click the label. So now it is showing the distances as well, that is 2.7, 2.2, 2.6, 3.2, etc, etc. But if you want to label the residues as well, I mean, you want to know what is this residue, what is the name of this residue, what is the name of this residue, there is another way to do this. I'm going to expand it a little bit for you so that you can have a good. Okay, so what you will have to do is just click on the residue, all of them. Okay, now just right click there will be a label. When you will right click into this interface, that will be residues. Okay, so now the residues has been labeled. This is phenylalanine, this is arginine, serine, proline, and along with their number. This is 308, 3112. So you can, this is the very good picture. So you can again go to here, file, and export image as PNG. So save image as PNG, image 2. So in this way, you can export all the or you can save all the images you want. Okay, so come here and check the images. This is image 1 that was in that specific orientation. You can use it for publication as well, and this is the image 2. Okay, so this was the only with the interacting residues. So now we are changing the format of we have hidden this show the ribbon or show the cartoon. Yes, again we have showed it again. So now how I showed it again, by clicking S for show and then cartoon, because we have hidden the cartoon as well. I am repeating it again for you. If you have made the interactions, how you will do it? Just go here, just firstly if you have shown the interaction and you want to hide the other parts, you will have to go to the H and hide cartoon. I have represented the ribbon as well, so I'll have to hide them as well. So yes, you have only the interacting residues. And if you want to show the ribbon structure, so by clicking S, click the cartoon. So now you have the complete complex. So I want to save the complex in the PDBQT so that I may be able to use it for some other analysis. So click the file and export molecule. From here, if you will save it, this is the, okay, so this is the format. I'll change the format from here, that you can change it to Maestro, you can change the PDBQT, PDB, MOL2, SDF, etc, etc. So I am going to change the format from PDBQT to PDB. So click here, the name is like one, yeah, or complex one. Okay, so I am saving it by the name complex one. Save it. So this is now saved. Where is complex one? This is complex one, and this is into the in the format of PDB. Now I am going to visualize it into the Chimera. What is Chimera? I have already told you in my previous videos. You can download the Chimera as well. Chimera, City of California, San Francisco. You can download the Chimera from here. Okay, so if you have Windows, you can select this one. If you have Mac OS, you can select this one. If you have Linux, you can select this one. So in this way, you can download the Chimera and then install it. So I am going to visualize my complex into the Chimera. So the name of the complex was complex one. Where is the complex one? this is into the PDBQT. Okay, so now you have shown this. This is the our ligand. Like if you click the select, then chain, then no ID, the ligand is selected. Okay, so you can also check the interaction in here. If you will click the tool and then structure analysis and then find hydrogen bond, and if you will apply it, so okay, oh my god, so yes, this is a hydrogen bond, that is with the histidine 239 and serine 308. So you can make the hydrogen bond over here. How you have shown the hydrogen bond? By clicking the tool, then structure analysis, find hydrogen bond and click include intra-residue hydrogen bonds. So you can change the color of the hydrogen bond from here. Like if I will click this or apply, the color will change. This is the hydrogen bond. You have changed the color, and you can show the distances as well. Label hydrogen bond distance. If you will check it and then apply, it will show you to the distances as well. Okay, so this is the distance. This is it is in very very small fonts. You can change the size of the font as well by clicking hydrogen bond. There are many things you can do this. I'll make a separate video on Chimera and how you are going to analyze the results in Chimera as well. So I will make a separate video as well. But in today's the clear selection, I'll show you something important. If you want to analyze it and you want to make a very good picture, so you can go to the action and then first select the receptor by select chain and then X. In my case, the name of the chain is X. In your case, it may be somewhat different. So this is not the rule of thumb. They may be different as well. So then after selecting, then go to the surface and show. So now it is in the surface format. This is in the surface. So if you want to change the color, like orange, okay, the surface is orange, and if you want to make it somewhat transparency, you can change. So now you can check this, it is 50%. okay, so this is the 50%. I'm changing the color to some magenta. Yes, now it looks beautiful. Okay, so if you want to save the picture by using the Chimera, set your the pose or the orientation in which you want to get the picture, like this, then go to the file, save image. Okay, here is something important, like I am going to the image, given the name image3. Here you can check the transparent background. So the background which was black in the image, it will be changed. Don't do anything, but by using just you can change here. the resolution, the DPI, print resolution. If you want a very high quality picture, just type it here 6000, or you may use 3000 as well, but I usually use the 6000. So click the save. Image is too big, make a smaller image. Okay, it is too big. Why it is showing too big? I'll just show you. Okay, I'm just giving 3000. I'm just checking what is doing here. Where is the images? No, it didn't make the image. Okay, so again I am clicking the image, and then here 2000. Why it is doing like this, I don't know. No, again. Okay, just do it 1000. Oh my god, what's the problem? I don't know. So you can use the prints. So okay, here $300. I'm going to do it 300. Okay, now it is making the image. So you can use 300, 600, whatever you want. So it will make a beautiful image for you in that specific orientation, but you can change it. You can change it whether according to your need or according to the protein, in which format you are going to use the protein structure, or if you have a protein ligand structure, so how you are going to use it. So it will be changed accordingly. This is not the specifically this tutorial is not specifically for to create the images. I'll make another tutorial that will be specifically for image creation or generation. So this is only for the analysis of your results. So we have analyzed the results in different formats. First we have analyzed in PDBQT, and then we have saved, or we have changed the format to PDB, and now we are going to analyze it into the PDB format. So we have different formats. Okay, it is taking some time. Why it is taking so much time? I don't. Yes, this is the picture. It is here, but this is not clear. While when it will be done. Okay, I think it is done. It's done. Yes, it's done. So you can check, it is 21 MB picture. So it is a high picture. If just check the quality while zooming it, there is no problem in the pixels. Very accurate. Oh my goodness, very good picture. Are you looking how much zoom you are doing, how much you are going to zoom it? Very beautiful. No pixel disturbance. No pixel disturbance till here. Very clear, very clear. So it is very clear. So you can save the image into this high quality. Okay, that will be depend on to the protein, because I have I usually save in save by doing the residues or the DPI here 3000 or 2000. So you can use 300, 400, whatsoever your protein is. It will be changed according to protein to protein. Okay, now the very important part, the format which you have changed into the PDBQT, sorry, from the PDBQT to PDB in the PyMol, that may have some problem while you are going to check this complex for the 2D plot on the online server. So I prefer to change the PDB format into the Chimera as well. But there is a way to save or to change the format again to the PDB. By click, you will have to click the select and then select all. So everything will be selected. I'm just okay. Just close this session and click. What was the name? This will be saved in your history. The protein or the complexes which you have opened, it will be saved here in the history. So if you will click here again, the protein will be shown again. So there's no need to import it again. Just click over that the name, it will be again open. So here click select and then select all. So everything is selected. You know, this is the ligand, the receptor that is selected. After after clicking the select all, go to the file and then save PDB. There is the option save PDB. Just save PDB. The name of the PDB is, what should the name? The name is new 2. The name is new 2. Okay, so you can like you can choose any name. That is Muz. My name is Muzammil, so I can use the name Muz or whatever your name is. So okay, let's save it by the name Muz, M-U-Z. Okay, save here. So we have saved it. Now we are going to analyze the results in the 2D plot as well. So we are going to do this on the online server. There is a server. The name of the server is ProteinPlus. You will have to write on Google ProteinPlus. Spelling is not correct. ProteinPlus. Okay. So type ProteinPlus. So this Zentrum website will be open. So click on this. So this ProteinPlus window will be open. Okay. So you can choose it. You can import here by choosing the by going to the specific folder or directory. So you can also import from here. Like, what was the name? The name was, what was the name of the PDB? The name was the Muz. So I am dragging the Muz to this PDB format. Okay. So you choose file, the Muz.pdb, and then go. Click go. So here, yes. So this is the ligand. This is our protein. And first of all, we want to get a very good 2D picture or 2D interaction diagram. So here at the right side, there are different options here. So you are interested into the PDB diagram, sorry, the 2D interaction diagram. So click here and then pause view. What we have did, go back, click onto the 2D interaction, pause view 2D interaction option, and here it will be shown pause view. So click on the pause view and here it is demanding the name of the ligand. What is your ligand? So just click on this ligand. The ligand is showing here. When you will click on here, automatically the name will be represented in here. So just click the calculate. Wait for some seconds. So this is the way you can have a 2D interaction diagram. Yes, this is very good, you know, very good diagram, very good image. So you can save or you can download it. This is the interaction between the serine 308. X is the name of our chain. Okay, so this is the phenylalanine 117, phenyl 157, 157X, 239, arginine 312, glutamine 304, etc, etc, etc. So this is the interacting diagram, 2D diagram or 2D image of your complex. So you can change the format from here by clicking here in PDF, SVG, PNG. I usually save in the PNG. So by clicking the download pose view image, it will be automatically downloaded. Yes, it is downloaded. So you can use this picture in your thesis or research paper. No, very good picture, very good image. Okay, the game is not over yet. We have another very important thing. There is the binding site detection option as well, that is do G sites score. So by clicking this option, do G site score binding detection, so click do G site score. There are some options. The pocket detection. This option will check the druggability of your ligand, how much druggable how much druggable your ligand is. You can check here. If you want to get the properties and some sub pockets and some other things, so you can select such option as well. So by just clicking the calculate, it will calculate the pockets or some other properties for you. So this is very important. These are the results which have shown the pockets or the receptor. So you can visualize it here. How I have changed. Yes, there is some it take some time to show the result over here. Meanwhile, we can do something over here. There are some options as well. We are waiting for these results, and meanwhile we are changing the format of this protein ligand complex. This is the protein ligand complex. We are changing something here. So first of all, there is the option for protein, that is into the this is in cartoon. So if you want to change it some other format or representation, you can change. Then there is surface. I'm not going to change the surface at this time. This is background, the black. So I am going to make it white for you. Okay, yes, these are the results. While clicking over here, this is the pocket. The first pocket, the very important pocket, which has been detected by the do G site scorer. That is the pocket where already your ligand is present. It means your ligand is present in very appropriate way. So you can change the you can check the volume, you can check the surface, the drug score, that is 0.8, the simple score, that is 0.61. So there are a lot of information. If you are interested in this, so you can note it down, or you can show these results, and you can download the total all the properties or the features by clicking the download over here. So you will be able to download all these results. Okay, so now we are this was about the druggability, or the 2D plot. Now we are, if you don't have the Chimera, or if you don't have the PyMol, and you want to analyze the protein ligand structure, and you want to make a very good, very high quality uh, image for your publication, so you can do it here as well. How you will, first of all, if you want to change the representation of the ligand, so this I am going to change it to the ball to stick. Yes, this is changed to the ball to stick model. Okay, so I have changed it, and I can also change the color. This is white, the mouse behavior, the resolution as well. So now I am going to show the surface. So this is the surface. I have showed you the surface. If you want to change the color, that is to magenta. So yes, you have changed the color of the surface to magenta. Or if you, this is the opacity is 100%. If you want to change, so this is the 50%, this is the 40%. So you can change that as well. So if you want the picture in this specific conformation with this surface, so you will have to click this here, the resolution, and the huge 6x. By clicking on this tab, the under the screenshot resolution, just click the huge 6s. So after clicking it, oh my god, oh my god, what is doing, what is happening? Yes, it is making the picture at very high quality of the pixels. So the after clicking here, automatically it will be downloaded to your download folder. So it is downloaded. So I have clicked here. Yes, this is the picture. If you will zoom it, zoom it, zoom it, zoom it, no problem in pixels. Zoom it and wait for some time, for some seconds. Yes, no problem in pixel. Very high quality image. You can use this image for your research paper or for your thesis as well. So if you will off the surface, so you can save the image in this form as well. Again, go to that 6sx. Yes, the image will be automatically downloaded with the high quality. Yes, this is the image, it is downloaded. So you can see in how what is the quality, very good quality, very good quality. So this is for those who don't have any visualizer or who don't know how to use the Chimera, PyMol, and some other things. So this is very easy to use. This is very easy to use. Okay, there is the another thing which we can do. In Google, type PLIP. Protein-ligand interaction profile, PLIP. When you will type PLIP, the protein-ligand interaction profile page will be opened. You can do that again. Our PDB file was MOS, so drag it over here. I have dragged the file over here, and then just click on the analyze. It normally takes some seconds or a minute. So it is analyzing the results. Yes. Okay. The result has been shown. All type of interaction between your ligand and the receptor, that is shown over here. This is hydrophobic interaction. These residues are making hydrophobic interaction with this distance, with the ligand atom 10, 158, with this distance, with the ligand atom 11. So these are the information regarding the hydrophobic interaction. These are the information regarding hydrogen bonds. These are the information regarding pi-pi stacking. So all sort of information will be available over here. So if you want to get the results, so you can download the results in RST format or in XML format from here. If you will click over here, the result will be downloaded automatically, and all these information will be present in your result, so you can use these information into your research paper. Okay, just click here for 3D view. Okay, I have clicked. So now you can see the interactions. Yes, these are the interactions. Okay, so you can download this picture as well by clicking the for 3D. So now you are viewing it in the 3D format. Okay, so these are the different type of interactions you can download to visualize the PSC format, that is the PyMol format, or you can download the visualize as PNG. If you will click over here, the PNG will be downloaded. So this image has been downloaded. And if you want to analyze into the PyMol, so click over here. That will be downloaded into the PyMol format. So you can open the download folder, and then if you will click this format, automatically it will be open here. Only by clicking, this will be open here, and then you can do those things again which I have shown you already, like you can label them, you can do whatever you want. Okay, by selecting all these, so you can do all such things which I have already told you. Okay, tutorial is too long, but it is I hope this will be very informative for those who have some problem in analyzing the results, or those who want to publish their research, their thesis independently. They can do all these things by themselves. And if you think it is good for you, and so you can like, subscribe, and share my channel as well. So the next thing is there is another thing, that is PDBSum. Type PDBSum and then click over here, Embl EBI PDBSum. Here this window will be open. So at the left side there will be the option of generate. Yes, click this. So you can drag your PDB, that was mus.pdb, over here. I have dragged it, and you will have to give your email address over here. And after giving the email address, you will click the upload. So by uploading, the window will be open, in which it will show your job has been queued. Okay, so and then they will send the result to your email address, like they have sent the result to my email address over here. It may take 2 minutes, 3 minutes, 5 minutes, 10 minutes, or whatsoever. So this is the email which I have received from the PDBSum, and there is the PDB code and the password, or the link. By clicking on the link, I can directly go to the results. Okay, so this is the protein. This is our complex. If you want to check the property of the receptor as well, so this is the Ramachandran plot. You can click the Ramachandran plot here, so it will show the results of your protein as well, the favored regions, the allowed the residue in the allowed region. Then you can check how much correct your PDB structure is of the receptor. But we are not interested in today's tutorial about the this study. We are interested to visualize the protein ligand complex or to analyze their interactions. So I'll click here on the ligand. So this is my ligand. Yes, this is the 2D plot of the lig plot. So you have seen such type of plot in the some paper or research paper or some thesis. So this is the plot. The lig plot is okay. What is lig plot? Lig plot is a software which is used to generate the 2D diagram or 2D images, or it shows the 2D interaction between the protein to protein and protein to ligand as well. But without installing, we can analyze, we can get the result or by the online server. Okay, so this is the way. You will have to just come to type the PDBSum, then generate, then you will have to upload your PDB complex, then you will give your email address, and then you will click to the upload, and then the link will be sent to your email. And by clicking the link, this window will open, and then by clicking the ligand, all these information will be available. So you can download the list of interaction from here. If you click over here, so this is the total details of the hydrogen bond, the interactions, the non-bonding contacts, etc, etc. These are all details. It is making one hydrogen bond, 79 non-bonding contacts. So you can report these things as well. And if you will click this PDB, so you will be able to visualize this 2D plot in the PDB format, and you can download it. So this was all about the protein ligand complex. And the last thing, there is another visualizer. The name is Maestro. So you can type Maestro free download. The Schrodinger website will be open. From here, this is Maestro free. Yes, here you can type Maestro free. You can download the free version of the Maestro. Like some people use the Discovery Studio as well. So the Maestro can also be downloaded in free version. I have saved it here. Okay, this is the free version of the Maestro. Here it is. Yes, you can download the version from here. If you have Mac OS, you have Windows, so by clicking the download, you can download accordingly what system you have. Okay, they have changed their website little bit. Free, you can search free Maestro here as well. So you will have to register first to their website on the Schrodinger, and then you will be able to download the free Maestro as well. Where is the free Maestro? I have bookmarked the page. So you can check to the Google, go to the Google, and you can download This is I think this is the page. Yes, this is the page, that is www.shringinter.com/free maestro /. Okay, so you will have to download, you will have to give the name over here and the password. After the logging in, there will be option of download, because you will have to register first for the Maestro. Then the Maestro will be downloaded, and then you will have to install the Maestro as well. So if you have installed the Maestro, this will be open like this. This is the Maestro interface. This is the academic version. So you can again drag your complex over here, and then here this is the ligand interaction box, so you can click. So this is a very good picture again for the 2D plot. So okay, okay, so these are the legends. You can go to the view and LID legend. So it will show you some different type of interaction. If this is this pink mean hydrogen bond, and this mean pi stacking, this green dotted line means pi stacking, and these residue are charged negative, blue are charged positive, green are hydrophobic, etc, etc. So and this like cyano are polar, and these are different type of residues around this ligand. So you can save this diagram or this image as well. Again, you will go to the save, then save screenshot, and here again you can click the transparent background, and then if you will make 6000, so it will make a very good, very high quality image. And if you will type 3000, it will again make a very good image. So I'm just typing 6000. You will have to type the width, the height will be automatically adjusted, and click the OK. So directory where you want to save it. Okay, I'm just going to save it onto the desktop. Name is Moz, and then save. Okay, now come to the desktop, name moz.png. So you can see, very beautiful, no pixel problem. Zoom it, no pixel problem. Okay, so this diagram or this image, this picture can also be used for publication. So this was all about from my side. By watching this video, you will be able to know how you can analyze the results, and how you will be able to generate high quality pictures, which you may use for publication or for your thesis. So take care, and yes, subscribe, like, and comment. Bye bye.